Hi, my name is Korbin Davis, and I'm here to talk to you about meiosis and mitosis. But first, let's talk about mitosis. Mitosis begins 6 steps. The first step is interphase. The cell spends most of its time in interphase than any other part. In interphase, the chromosome makes an exact duplicate of itself, and the chromosomes thicken and coil. Then prophase. In prophase, the centrioles divide, form asters, and move apart, as the nuclear membrane begins to disintegrate. And as soon as prophase begins to stop, the central and asters are at opposite poles. The nucleus and nucleal membrane have almost disappeared. In prometaphase, the centrioles spindle fibers begin to attach to the chromosomes and begin to pull them towards the center for the start of metaphase. At this time, the nuclear membrane has almost disintegrated. After that step is metaphase. In metaphase, the centrioles pull the chromosomes to the metaphase plate. Then you start anaphase. In anaphase, the centromeres split. Half the chromosomes move to one pole, half move to the other pole. As anaphase begins to stop, the chromosomes have almost reached their respective poles. The cell membrane begins to pinch at the center. If this is an animal, this is called a cleavage furrow. Then we start telophase. The cell membrane completes construction in telophase. The nuclear membranes form around the separated chromosomes. After this, mitosis is complete. There are 2 cells with the same structure, number of chromosomes, as the parent cell. After mitosis, let's talk about meiosis. Meiosis is how we create sperm and how ladies create eggs. Meiosis consists of interphase 1, prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, and telophase 2. Interphase, just like mitosis, is where the cell spends most of its time. Then it moves to prophase. Prophase, DNA strands thicken into chromosomes, homologous or like chromosomes, began to approach each other. The homologous chromosomes pair to form bivalents. Centrioles divide and move to opposite pole sides. Then, the bivalents duplicate to form tetrads. The nuclear membrane disintegrates, crossing over occurs. Then it becomes metaphase 1. In metaphase 1, the tetrads attach to spindle fibers at their centromeres and line up mid-cell. Then anaphase 1. In early anaphase 1, the tetrads separate and their paired chromatids move along a spindle to their respective centrioles. In late anaphase 1, the chromatids have almost reached spindle poles and the cell membrane begins to constrict. Then, in telophase 1 in cytokinesis, the nuclear membrane encloses the separated chromatids. The cell membrane completes its constriction. The first melodic division ends. There are now 2 cells, each with the same number of chromatids as the parent cell. Then you go to prophase 2. Prophase 2 begins in the second meiotic division. Homologous chromatids do not duplicate but merely separate. Then you go to metaphase 2. The chromatids in each cell line up mid-cell. The centrioles and asters are at the poles of both cells and a spindle has formed. In anaphase 2, the now separated chromatids approach their respective poles. The cell membrane begins to constrict. And then after this happens, you go into telophase 2 in cytokinesis, and telophase 2 has then been completed. There are now 4 cells, each with half the number of chromosomes of the parent cells. Each cells are haploid. They are not diploid. Each cell creates what us humans call sperm to reproduce. Thank you for watching, and I hope that you like this little video I made.